Hello again. Welcome to this Aston Originals podcast. This is the next in the series of the AIM podcasts entitled What is a Membrane? AIM is the Aston Institute for Membrane Excellence. I'm Dr. Matt Derry. I'm a senior lecturer here at Aston University, specialising in chemistry, polymer chemistry. And I'm joined today by Dr. Doug Browning. Hello, welcome, Doug.
Thank you very much for having me.
Doug has been at Aston for almost 3 years, and you're a senior lecturer in biosciences here. You're an expert in molecular biology, and quite interestingly, you described that to me as being an evil genetic modifier, but I'm sure it's not even that's the case. We'll find out about that. And it's engineering life to do useful and important things, and that's exactly what we're going to be discussing today. And it fits within the engineering biology sector, which is a priority for the UK government and lots of research going forward in the biotech sector. So it's really exciting to have this project. And the project is a BBSRC Engineering Biology Mission Award, which was awarded, and the project started in April this year. It's a 2-year project with Doug here at Aston, and also Professors Alan Goddard and Rosalind Bill, who you'll have heard from AIM as well, in conjunction with 2 researchers from Warwick, Dr. Alexander Darlington and Professor Phil Stansfeld. So it's a big grant, £1.76 million, which sounds very, very impressive, and I'm sure it's really exciting work. So can you tell us about the project and what the project motivation was, please, Doug?
Yes, thank you very much. Yes, it is a really exciting project, and thanks for having me on today to talk about the work. So in a nutshell, what we're trying to do is we're trying to make ecoli cells, bacteria, express large amounts of proteins, so that of membrane proteins, so we can use this to carry out various important sort of research and investigations. So, I mean, one of the key questions is, well, why are membrane proteins important? And I'm sure you're aware that most cells, well, in fact, all cells have membranes surrounding them to keep all their contents in. But the important thing is that cells have to allow things in and out of them. Okay, they have to communicate with each other, and they perhaps have to form attachments between each other. And this is all carried out by membrane proteins, and this is why they're so important. And they're essential for life. So without them, we wouldn't exist and life wouldn't exist. It also makes them really important for medicine and biotechnology, because these are often important targets, maybe for designing drugs to cure diseases, or to make vaccines to protect people against pathogens or viruses and bacteria. The important thing about membrane proteins actually is they're present in very large numbers in cells. So about 20 to 30% of all proteins in an organism is a membrane protein. So they're important. But the problem is they're often there in very small amounts. And they also love to live in the membrane, which means that they like very sort of hydrophobic or lipid-like environments. So they're sometimes very hard to extract and work with, and especially to get enough protein. And so this particular mission award that we're on is all about trying to make cells express more protein so people can work on them and perhaps discover more drugs that might target important membrane proteins that are involved in disease and other sort of factors.
Amazing. So yeah, that was, for me as a chemist, a really good introduction to what membrane proteins are and why they're so important. And it feels really important to be able to get enough material to do something with. So how did you come up with the idea for the project?
Well, it's been a long-standing problem, and it's for many researchers, actually getting enough protein and getting it out the membranes is quite difficult. And what you tend to find is sometimes when you express a protein, the cells don't particularly like it. When you want to express your proteins, you want the cell to be happy and healthy. But often, when you try and express large amounts of protein it's not used to, this triggers what we call stress responses. So this might be something simple like the cells, because they're producing so much membrane protein, they're not producing their other components, they might enter starvation. The other alternatively is that they might not be able to fold their proteins properly, and so they then have sort of a folding stress. And this can all lead to protein expression causing your cultures, this is what you're growing your bacteria in, or your sort of cells like insect cells, mammalian cells, to stop producing protein, and they might stop growing, and you want the most yield of your protein. The problem is, for each kind of protein you do, you have to tweak this. So it's a very sort of what would you say? It's a very hit and miss system where you have to optimize the expression conditions for each protein that you use. And what we wanted to do at the start of this mission award was to make a system that actually would work out the best conditions to express your proteins. So what we wanted is we wanted sort of a self-regulating system, a system that was clever. So when the system realized, when the cells realized that they were causing stress, they can switch down the production so the cells are much happier. And I like to think of this perhaps as when you're trying to cook rice on a stove. So you've got your pan and you pop it on the stove like that, your rice goes in, and you put your boiling water on. Turn the hob on and off you go. You turn around, chop some vegetables, something like that, and come back and you notice that the pan's almost boiling over because you've put too much energy into it. You've tried to produce too much protein. And at that point, you rush over and you try and shut it down. But it'd be nice to have a system that would do it for you. And that's essentially what we want to do. We want sort of a cooker that actually controls, so you can just have your rice simmering nicely and cooking while you're doing something else. So that's essentially what we're trying to engineer into E. coli. We're going to make E. coli clever, so it'll have a smart, autonomous, regulated system. So when the cells are producing protein, if they start to produce too more, they turn it down, and so they can grow to very high levels and produce lots of lovely protein for everyone to work on.
Okay, so a rice cooker to keep your cells less stressed.
Exactly.
So they can produce the best quality membrane proteins and more of them.
And in some ways, it's not just about producing lots, it's also producing about active protein as well. So often it's not having huge amounts. We want to do things with it, and often we'll have to look at how these proteins work. So they must be active, and that's really important, that they're folded properly and they can carry out the job they're meant to.
Great, thank you. So you mentioned membrane proteins and their roles in vaccines and even drug targets. So what is that role? What do membrane proteins do?
Okay, so what you find with quite a few membrane proteins is that they're often involved in disease processes. And the best thing would be able to come up with a small molecule that can perhaps stop the disease occurring. So perhaps one of the classic examples is what we see in cystic fibrosis. So cystic fibrosis is a genetic disease. And what happens in individuals in that case is that there is a mutation in an important transporter called the CFTR transporter. In some instances, this completely destroys the protein, but in other instances, it makes it work poorly. And this particular transporter is involved in transporting ions in the lung. So from out the cells into the lung. And that's what makes the mucus in the lungs all nice and sort of, I guess, liquid. But if you have a mutation, what it means is that the mucus in the lungs becomes very sort of sticky, and people find it very hard to clear this. And what then happens is bacteria like Pseudomonas are able to colonize the lungs and start to cause disease. Okay, and this isn't good for the individual, and it often means that if they get a severe infection, it can be life-threatening, and it does affect people's lives considerably. So what drug makers have started to try and do is that in instances where the CFTR transporter doesn't work so well, they've been designing small molecules that can interact with the receptor, with the transporter, and allow it to do its job. So it can then transport ions into the lung, and the water will go into the lung, and therefore the mucus is how it should be. And this can actually be a great sort of life-changing moment for people with cystic fibrosis. The other thing that membrane proteins are really good for is obviously for vaccines. So the idea of a vaccine is what you do is that you give an individual a protein that comes from a pathogen. So this might be a virus or a bacterium that causes disease. And because you inject it into them, their body has seen this protein, and it means that it can start to mount an antibody response towards this. And then if you happen to get an infection with that particular organism, your body is already primed to attack it. Okay, so it means that you have immunity, you're less likely to get the disease, and in fact, you'll probably clear the organism much more quickly. And so that's been really important in vaccine science for many years. There are lots of top-selling drugs, vaccines that use membrane proteins, and perhaps one of the most important one is the COVID-19 vaccines. So they often contain the spike protein that's part of the COVID virus. And this means that you can raise an antibody response to it, and it'll stop the virus entering your cells and causing COVID. There's other things such as the hepatitis B vaccine, which also uses a surface membrane protein. And then things like the whooping cough vaccine that we give our children, which is really important. A component of that is actually an outer membrane protein from the bacterium that causes that. So you can see how membrane proteins can be very important to target in medicine, and also very important to generate immunity and keep individuals healthy when they're exposed to quite severe pathogens.
Brilliant, thank you. So if we weren't sure about the importance of membrane proteins before, I'm sure we are now with those brilliant examples. I want to go back to this rice cooker. So you mentioned if it starts to boil over, in the analogy at least, you turn it down so you can control the process a bit more. In the context of membrane proteins, how does that work exactly?
So how are we going to make this self-regulating system? So obviously we've got a great collaboration with Alex and Phil at Warwick. And the idea is that what we're going to do is we're going to use both computer aided modeling and also what we call wet lab work. So we're going to work in the lab on the stuff. So the idea is the computer modeling is going to be carried out by Warwick. And in this instance, what I mean by this is what they've built in their computers is sort of a virtual version of a cell. So you can sort of alter parameters and see how this virtual cell operates. So the idea is what they're going to do is they're going to have their virtual model, and they're going to induce essentially recombinant protein production. And from this, they're going to try and work out which pathways are stimulated and which stresses perhaps take place. And by this, they're going to try and ensure that their virtual cells grow very nicely, okay, so they're not too stressed, and that they can produce most active product they can. Okay, so they will then come to us and say, well, look, these pathways seem to be important, these stresses are important. And what we're also doing at the same time is trying to work out which stresses are switched on. Okay, so that's the idea, is that we will then use the computer modeling and also what we've learned to then create systems where our expression systems will have certain tweaks to them, that when a stress switch is on, sort of the light bulb is turned down slightly and you don't get much production of membrane proteins. And so that will try and avoid the stresses. We'll have cells that grow properly. And more importantly, they'll be producing protein that is folded and active, okay, into the right lipid environment. And then that's very important. If you want to go on and do a drug screen, you've got to have active protein. So the idea is a combination between computer modeling and actually wet lab work to design a new system that becomes smart and essentially turns down your cooker when you want.
Great. So this is that evil genetic modification, but for good.
For good. Well, yes, I like to think of it more as sort of your friendly neighborhood.
Okay, we'll start
genetic modification.
Yes. So you made it sound incredibly simple, which I think is great, because hopefully, well, the lowly chemist can understand what's going on here, at least as much as I need to. Is the process as simple as you optimize for one membrane protein and then you can do it for another, or are there intricacies between different systems?
So that's really important in the fact that often a membrane protein, perhaps from an insect cell or a human cell or a bacterial cell, they need certain lipids. They need certain types of lipid in their membranes to help them work. Okay, so just producing huge amounts of protein might not be effective because you haven't got the right lipids. So one of the other things we're trying to do and we're focusing on using harmless E. coli here. So we're not being evil in any way at all, just to reassure everybody, is to try and make E. coli produce certain lipids that make it perhaps more like a human cell or more like an insect cell. So what Phil is doing at Warwick, he's taking our proteins that we're going to express, so all the targets, so for example, aquaporin 4, and various other transporters. And he's going to model which lipids are important. Okay. And then in conjunction with us, what we're going to do is then we're going to tweak the lipid production pathways in E. coli. So they produce certain lipids that are favored for that protein. So we'll have a system where perhaps if you're producing a human protein, we might have an E. coli that's a little bit more, produces a membrane a little bit more like a human cell, and therefore any proteins we express are actually going to be active. So often lipids are very important in making them, not only keeping the proteins folded, but also in their activity. So it's very important to make sure that the lipid environments are exactly the same.
Great. So people might hear this and hear you talking about E. coli and think, obviously, they only think bad things when they hear this. We want to avoid it typically, but it's using it for good here. So it's a route to membrane proteins that can be useful for a wide range of things.
Yes. So, I mean, the important thing about E. coli is that, yes, we hear about the things on the news about E. coli 0157, which can cause disease when you perhaps have an undercooked hamburger. Most E. colis are, in fact, actually harmless. They're what we call commensals, that they live in your gut, and they actually provide and are helpful for you. The ones we tend to use in the laboratory have been used in the laboratory for over 100 years. They're very safe, and they're great sort of cells to express proteins in safely. So what we're doing is it's all about being safe and using the systems that have been established over a number of years.
Okay, you've convinced me you are the friendly genetic modifier. So using this platform technology to generate membrane proteins of a wide variety for lots of different applications. So I can see it being incredibly useful for many scientists around the world in academia, industry, biotechnology sector. So I'm personally very excited about this. It's great. Also, I guess the grant doesn't just pay for the research, it pays for researchers to do it as well. So can you talk a bit about the people who are employed on the grant?
Yeah, so at the moment, so the whole grant itself was to employ a number of researchers. So for example, at Aston, we've got 2 postdoctoral researchers, one working on the genetic circuits, so trying to work out the rice cooker, and the other one working on the environment. And we've also got the membrane environment. We've also got a research assistant that's helping on that. So together, that team is working to engineer the cells. And then at Warwick, there's also a postdoctoral research assistant working on how we can work out what stresses are induced and then another one working on the membrane sort of environment and modeling that, how membranes, proteins interact with lipids. So that's one part of the grant, but there's other things that we've said we'd do. So for example, obviously, we're really looking forward to interacting with various industrial partners, and there are quite a few involved in this. So one of the things we're doing is ensuring that all of the researchers have some entrepreneurial training. So we've got some nice business courses lined up. And we've also want to share what we generate with the public. So we're going to be setting up a 2-day symposium where we have invited speakers that talk about their research, but we'll also be telling industry and also the scientific community what we've done as well. And coupled with this, we're having sort of a lunchtime seminar series that will be online, so we can put up more details about that soon. Excitingly, what we've been given by the BBSRC is a little bit of additional money to try and work out if we can make our new systems, and the systems we already have that we're building on, if we can use those to perhaps set up a spin-out company. So we're obviously looking into marketing and interacting with particular industry partners to see if we can become a company where people will come to us and say, we're having problems expressing this protein, what can you do for us? And so we'll put it into our new smart strains with our new lovely lipid environments and hopefully express large amounts of active protein for them, and then take the technology that we've used to actually do some good and allow people to discover more drugs or investigate membrane proteins that are particularly important in medicine.
That is incredibly exciting. So
Yeah.
I guess my message to anyone who's in industry or needs to have a lot of active membrane proteins, get in touch, come and use Doug's rice cooker. You can keep the cells nice and relaxed, not stressed, and then optimize this process. Amazing. So that kind of wraps up today. Thank you so much
Thank you
for
for having me.
talking about this great project. Check out more information to come via the Aston channels, certainly on the AIM website as well. AIM itself, I'd like to thank Research England for funding. So we started as an official institution in August of 2024, so just a couple of months ago now. And we've got some momentum. We're doing some really cool things. Doug is just one great example of an active project of stuff happening in the general area of membranes, whether it's membrane proteins, polymer membranes, which is more like what my expertise lies in. So keep on track with all of the stuff coming out of AIM. Check out the website. We'll put a link to that when we release this as well. So thank you very much. I've been Matt Derry, and I've been here with Doug Browning,
Thank you.
and we'll see you next time.